Hi, good morning or good afternoon, depending on whichever part of the country or world you're in. My name is Padma Sundar, and I am the Director for Cancer Applications at Affymetrix. Today I'll be speaking about the OncoScan assay for copy number from FFPE and sharing several publications and clinical cases on actionable copy number detection using this platform. What I would like to cover in this talk are important publications highlighting the importance of copy number in several solid tumors, and then go on to describe the OncoScan assay, which is based on a proprietary molecular inversion probe technology that is especially optimized for assessing copy number from highly degraded FFPE samples. And then I will go on to describe some important publications on the research side, but as well as some important clinical cases on the clinical side that demonstrate the clinical utility of copy number detection, of copy number in the detection of actionable alterations. First, I would like to bring to your attention a very important paper published in Nature in November 2013 by Ciriello et al. These were the results from the TCGA genomic profiling of over 3,000 samples in 12 solid tumors. These samples were profiled for mutations using NGS methods, for copy number using Affymetrix's SNP 6.0 technology, as well as for methylation patterns. The remarkable finding from this analysis was that it revealed the existence of two mutually exclusive classes of solid tumors. The M-class tumors that were driven by somatic mutations, but a completely separate and mutually exclusive class of tumors called C-class tumors, which were driven not by somatic mutations, but by copy number changes. The C-class tumors identified in this paper are ovarian cancer, breast cancer, squamous cell lung cancer, head and neck cancer, and as well as prostate cancer. And therefore, this paper really highlighted the importance of copy number and showed that copy number analysis is vital in the analysis of these C-class tumors. Digging a little deeper into this data, what you see here on this slide are the tumors along the bars, and on the top you see the M-class tumors, and on the bottom you see the C-class cases in terms of % of cases with an actionable alteration. On the extreme right, what you see is ovarian cancer in red called OV, and you can see that it's almost completely dominated by copy number changes, and they've rarely found mutation events in the top half. Right next to it, you see BRCA, which represents breast cancer, and again you can see here in the bottom half that over 90% of the cases had an actionable copy number change, but a far fewer number of cases had an actionable mutational change. Right next to it, you have squamous cell lung cancer, followed by head and neck cancers. And these were the cancers that were classified as C-class tumors. So now it's pretty well understood that especially for the C-class tumors, it's important to detect copy number. But the detection of copy number can be pretty challenging in solid tumor samples. As we all know, it's well established that it's very challenging to generate reliable DNA information from FFPE solid tumors. Next generation sequencing technologies have addressed this challenge for somatic mutations by the targeted sequencing of single base pair or limited base pair hotspots at a very high depth of 500x or 1000x. But copy number changes affect larger regions, sometimes genes and entire chromosomal arms, and sequencing depths across those regions tend to be much lower, which affects the results you get, resulting in false negatives. This is a snapshot of, this is from the Frampton paper that describes the validation efforts of a next-generation sequencing technology that sequences over 300 genes at a 250x depth. And even at this high depth, it acknowledges gaps in copy number detection. So what you see here are important markers, which could be predictive, prognostic, and diagnostic along the column, and gaps in the detection of copy number events, which could be amplifications, deletions, or copy neutral LOH. What you see here that even according to the paper, even at 250x depth, lower level amplifications, meaning any amplification that is less than 8 copies may be missed. The other important category that is missed is hemizygous deletions. And of course, it's very challenging at 250x to detect any copy neutral LOH. So amplifications that are below 8 copies, hemizygous deletions, and copy neutral LOH events continue to be a problem even at 250x coverage according to this paper. The next logical question is, do these lower level amplifications matter? Do amplifications below 8 copies even matter? Here is an actionable, well-known actionable copy number change, which is the MET amplification, and a paper by Capuzzo et al. That highlights that the cutoff for patient selection for a MET amplification is 5 or fewer copies, so below the 8 copy threshold. And this is a very well-established actionable gene for copy number. A second example is from clinicaltrials.gov, where the recruitment criteria for several drugs for FGFR1 amplification again is 5 or higher copies. So the cutoff is well below the 8 copy that is possible with high depth NGS. And of course, the most well-known example of all is HER2, where the cutoff is 4 copies for absolute copy number and a 2.2 CEP17 to HER2 gene ratio for therapy selection. And therefore, it's pretty well established that it is important to detect lower level amplifications at 4, 5, 6, and 7 copies. This is the Affymetrix developed the OncoScan assay specifically to address the gaps in current technologies. We just talked about sequencing, but I just want to briefly talk about other methods, such as other microarrays. The problem with other microarrays is that they are not optimized for FFPE, with the result that they require a large amount of input DNA. So other array CGH technologies require 500 nanograms, which can be very problematic for the FFPE sample type. They are unreliable, so several of these assays require DNA restoration as a first step. But even after that, the quality of the data can be unreliable. And unfortunately, there are no QC steps that can tell you ahead of time whether the end product is going to be noisy or usable. Which means that until you have consumed the entire precious sample and spent the money and labor on the entire assay, there is an element of risk in terms of whether you're going to get good quality data at the back end. FISH is widely used for the detection of copy number, but it's obviously very locus dependent, and as I'll go on to show you, as more and more copy number changes are deemed actionable, FISH becomes impractical. And of course, for research purposes where users are looking for a whole genome approach, FISH is not a useful technology. And this is the reason that Affymetrix developed the OncoScan FFPE assay kit for the analysis of solid tumor. The OncoScan FFPE assay kit is for the genome-wide copy number profiling of FFPE samples with results, meaning you go from DNA to your actual copy number calls in 48 hours. So just before I get into the technology, which is actually the reason to believe, I'd like to share some high-level product specifications. The OncoScan FFPE assay kit only requires 80 nanograms of FFPE-derived DNA, but it's been validated down to 50 nanograms. It's a quick assay, meaning you can go from your extracted DNA to your copy number answers genome-wide in 48 hours. It's whole genome, which obviously makes it very useful in the research setting, but it also has high resolution in 900 well-characterized cancer genes, which makes it very appealing in the clinical setting as well. It also enables loss of heterozygosity detection of less than or equal to 10 megabases across the genome. And it's a highly sensitive technology, meaning you can detect down to 3 copies present in 25% aberrant cells. A very important feature in heterogeneous solid tumor samples where only a fraction of the cells might actually contain the aberration. Unlike other microarrays, this comes with a solid tumor algorithm that actually calculates the copy number in the tumor portion only, subtracting the contribution of the normal diploid cells to the copy number. It has a dynamic range of 50 plus copies, and you get linear copy number calls all the way from 0 to 50 plus copies. I'll go on to describe its performance in beta testing, but 99% of FFPE samples that were tested generated high quality data. And because of the nature of the molecular inversion probe technology, it can be used on archived samples, which a lot of researchers really love, because they can go back now and conduct retrospective studies on older samples on which they have clinical trials and generate data with a high pass rate and get to their publications and signatures very quickly. So it's been validated on archived FFPE samples that are up to 20 years old and highly degraded FFPE samples that are less than or equal to 100 base pairs. And several studies have been conducted demonstrating concordance and superiority compared to FISH and array CGH methods, which are available on our website. So we were very privileged to have been selected by 3 prestigious labs for the beta testing of OncoScan. These were the ARUP labs, MD Anderson Cancer Center's Diagnostic Lab, and Memorial Sloan Kettering Cancer Center's Diagnostic Lab. And the way they tested the performance of OncoScan was to run samples with known FISH status and look at 2 things. One is, what fraction of the samples generated high quality data on FFPE? And secondly, what was the concordance compared to FISH? So as you can see here from this little table, 99% of the samples generated high quality data on FFPE, and there was excellent concordance with FISH in most cases. The one interesting thing was that the OncoScan actually uncovered 3 false positives in FISH in 3 cases, and these have been published. What happened in these cases was, because the FISH method, these were all HER2 cases, and as you know, oftentimes in FISH, what is measured is the ratio between the CEP17 probe in the centromere and the actual HER2 gene. And when the centromere has a deletion, that can skew the ratio and make the HER2 look like a relative gain, even if it's normal. And this is exactly what happened. And this is how the false positives were uncovered on OncoScan. In each of these cases, the centromere was deleted, so the ratio of HER2 to CEP17 looked like a positive, but the HER2 gene itself showed no amplification. And because OncoScan is a whole genome product, we were able to see both the centromere region and the HER2 and able to determine that this was not, in fact, a true amplification of HER2. And this is the reason why MDL and other labs have one use case of OncoScan, which is around validating calls where there is a discrepancy between FISH as well as IHC expression in these cases. So what is this product about? It's a copy number panel of 220,000 probes, and all of these are SNP probes. The advantage of having a SNP probe is twofold. A, it gives you 2 data points per call, unlike other array CGH methods. You see a shift in the log-to ratio, and you see a corresponding change in the SNP pattern. And with these two data points, it gives the user the confidence that the call is a true call, meaning routine labs, once validated, are not confirming, are not needing to spend the money or sample to confirm OncoScan calls with a secondary method. And the other advantage of the SNP is that even when this log-to shifts are subtle in low-level mosaic calls, you get that splitting of the SNP bands, which gives you added confidence that even though the log-to shifts are subtle, the call is real. Again, 2 data points per call. Like I said, it's a whole genome product, but very high coverage in well-characterized cancer genes. And these genes were selected using the Sanger and CCMC databases. And you have very high exquisite probe density on those at the range of 1 probe to 2 or 5 kb, which allows a very high resolution of at least 50 kbs on these genes. But then beyond those 900 cancer genes, you get genome-wide resolution of 300 to 380 kb. Now, I want to describe to you the technology behind this that makes this platform so conducive for degraded FFPE, and that's called the Molecular Inversion Probe technology. Before I want to remind the listeners that although the current OncoScan kit in its kit form is new, this is a proven technology, and this has been offered via Affymetrix's research labs for the last few years, and it's a proven technology with over 80 publications in the last 2 years, and over 12,000 samples have been processed with a 93% pass rate in FFPE. So the technology is proven, and it has had several publications in the last couple of years. So let me describe the technology by starting out with what we design. What we design here is the following probe. What you see here in red are sequences that are complementary to the sequence of interest that we are interrogating for copy number. In black, you see classic or standard PCR primer sites. And then in green, you see a universal tag, which is used only to read out the sequence that has already been captured. So the intelligence is all happening using the red sequences, and the tag, which then hybridizes to the microarray, is simply a readout. So this is how it works. You can see the genomic homology sites, you can see the PCR binding sites, and you can see the tag array. What happens in the first overnight step is you add these 220,000 probes to your genomic DNA, and in an overnight annealing step, the genomic homology regions bind to the complementary sequence of interest on the genomic DNA. In the morning, this process has been completed. So this is the process where it's binding overnight. And in the morning, what you do is you come back and you add your DNTP, ligase, and your gap fill, and that's the genotyping step. In the previous night, the annealing step has happened, leaving one open base pair for the genotyping step. And when you add the DNTP, when it finds the right complementary DNTP, that gap is filled, and now what you get is an airtight, closed structure called the Molecular Inversion Probe. At this stage, you do an exonuclease addition steps, which is very important, because what it does is it gets rid of the extra probes that were not, for whom the matching sequences were not found, and it also gets rid of the extra probes that were not found to be, that were not annealed to the genomic DNA. So now, after that's been done and you've got rid of all the unwanted DNA and the unused probes, a cleavage step occurs and the linearized probe is inverted, and hence the term inversion probe. And now, as you can see, you are left with just the correct probe sequences that found the matching genomic DNA. And now these linearized probes with the universal primers are ready for amplification. And this is when we do the we do the amplification. So you can note that unlike other assays where you start with PCR or genomic DNA leading to bias, here there is no genomic amplification, and the only thing that's amplified is the correct probe sequence that has found its complementary sequence on the genomic DNA. And this is the reason that you get clean, unbiased results. What I also forgot to mention is in the first step, the genomic homology regions that you look at are only 40 base pairs. And therefore, this is highly optimized for degraded DNA because all it needs is 40 base pairs of intact sequence. So back to the step where we are now left with a lot of amplified probes, correct amplified probes. We end up with a HA3-mediated restriction digest at the second cleavage site, and now all you're left with are the tag sequences, and following that is a classic hybridization step and then the final readout. Again, this technology is conducive even to eventually to even sequencing, because as you know, as I showed you, the detection is all happening in the annealing step, and this last step on the array is simply a readout. And now again, going back to the summary, because of the way the molecular inversion probe technology works, the first point being it only needs a 40 base pair sequence. The second point being there is no genomic DNA amplification, only the correct probe sequences that have found the matching genomic DNA sequence are amplified, is why this technology is optimized for degraded DNA, and this technology gives you clean, noise-free results from FFPE. And this has been acknowledged in several important publications, and I will be showing you one of them shortly. This is an important publication that I'd like to highlight that talks about how few studies have realized the copy number potential for FFPE with damaged DNA because of the requirement of large amounts of genomic DNA when it comes to other array technologies, or very high depth sequencing coverage and the subsequent added bioinformatics challenges that this brings. OncoScan, based on molecular inversion probes, according to this publication, appears to perform well in comparison with array CGH platforms, and so might prove to be a successful method for studying FFPE samples for copy number alterations. Just as a reminder, if you like, at any time you can submit questions into the Q&A box, and I'd be happy to answer it at the end of the talk. Thank you. So now I will segue into the application part of this OncoScan assay, sharing with you some important publications on the research front and some important case studies on the clinical front. We all know that biomarkers can play the following roles in cancer. They can be used for patient diagnosis, which is simply to determine whether a patient has cancer or not. Post-prognosis biomarkers can be used for prognosis, which is simply for a diagnosed patient, estimate the likelihood of recurrence, metastasis, or death. The idea being that you treat patients with bad prognosis aggressively, and you wouldn't overtreat patients with good prognosis. And finally, the third application would be therapy prediction, where for a diagnosed patient, you predict whether a treatment or drug will, a patient will respond to a particular treatment or drug, or equally importantly, you'd want to know markers of therapy resistance, meaning will a treatment not work in a subset of patients who have that biomarker. For OncoScan, it's being used in 3 different segments. One is the research segment, where it's being used to discover novel biomarkers that are of prognostic or predictive value. It's being used in the pharma segment, again in the biomarker discovery world, to discover and validate novel predictive biomarkers and use those biomarkers for trial patient selection. And finally, it's being used in the routine use setting, where the idea would be to consolidate multiple markers for diagnostic, prognostic, or predictive value. So one of the important papers for biomarker discovery that I would like to highlight was published in Oncogenesis, a Nature publication. And this was a study conducted by Genentech, where what they did was they did a whole genome scan on several breast cancer samples, and they were able to identify and validate a novel biomarker, which was an acquired PI3 kinase amplification that causes resistance to selective PI3 kinase inhibitors in breast cancer. And this was obviously a very important finding given the number of PI3 kinase and mTOR inhibitor trials that are going on now. So this was a case example where they were able to, because they did not know what they were looking for, they were able to leverage the whole genome scan using OncoScan and then narrow it down to the single marker of therapy resistance, and then perform a functional validation of that PI3 kinase amplification using PCR-based approaches. And this obviously, like I said, has significant implications for trials, both PI3 kinase and mTOR inhibitor trials. This is not the only example. There are about 12,000 publications based on the OncoScan-based MIP technology, and you can find more of these on our Affymetrix website. So far, about 12,000 samples have been processed on this technology with a 93% pass rate in FFPE. I'm just highlighting some recent publication on the OncoScan technology from 2014. As you can see, they do over-index on C-class tumors that I described earlier in the talk, but they span a large number of tumors here, including several brain tumors, colorectal cancer, bladder cancer, fallopian tube carcinoma, and so on. So this technology, being genome-wide with high resolution and 900 cancer gene and being compatible with any degraded DNA, can really be used for any solid tumor research application. But the use of the OncoScan FFPE assay kit is not just limited to the research setting. Leading routine use labs are also rapidly adopting OncoScan for copy number. At this point, there are 30 US labs that are trained and finalizing implementation of OncoScan for routine testing. Memorial Sloan Kettering, ARUP Labs, Fox Chase Cancer Center, and Seattle Cancer Care are offering OncoScan in a lab as a lab-developed test in a CLIA environment. The OncoScan FFPE assay kit has also received New York State approval for DNA copy number and allelic imbalance analysis of solid tumor tissues using the Affymetrix OncoScan SNP array assay. And this was received within 6 months of us making the kit available. So how are these routine use labs using OncoScan? These labs will combine somatic mutations from existing NGS panels for mutations with OncoScan for copy number detection. Now I would like to, in the final few minutes of the call, I would like to share several case studies in the routine use setting where by adding copy number from OncoScan to somatic mutations, the users, the oncologists, were able to both increase the number of cases that had an actionable alteration, and secondly, increase the number of actionable targets or therapies available to them. There are a number of possible definitions for actionable CNV. So for the purposes of this, the rest of this call, I'd like to define actionable CNV as defined as a generally accepted definition of what's truly actionable. So for the purposes of the rest of the discussion, an actionable copy variant would be one where the patient would be A, either eligible for an FDA-approved therapy, or B, would be eligible for a therapy in an open clinical trial that is currently recruiting patients. So with that definition in mind, let's look at some of these case studies on OncoScan. And also, I'd like to point out, this comes up a lot, that according to the Ciriello et al. Paper, there are at least 23 genes that are actionable for copy number meeting the definition of actionable discussed in the previous slide, meaning for these 23 genes, there's either an FDA-approved drug or a drug in clinical trial that can be given to patients who have this copy number alteration in one of these genes. And what the paper also points out is the pan-cancer actionability of these copy number. And there is a very new Cell paper, if you've not seen that already, that also talks about how the molecular profile of a tumor may actually matter more than the tissue of origin. So shown here, it's a bit of an eye chart, is that these 23 actionable copy number changes are found in a variety of across solid tumors, and they're actionable when found in these solid tumors, is what this simple slide shows. So now I want to highlight a Huntsman Cancer Institute study, courtesy of Dr. Josh Schiffman, an oncologist at the Huntsman Cancer Institute, and Dr. Sarah South, lab director at ARUP. This was a study that has enrolled relapsed cancer patients, which would be the category to first address, because oncologists typically are willing to go off protocol for these patients because their prognosis is so poor. So at first, what they did was just focused on somatic mutations, and they ran a sequenome panel of 277 mutations in 25 genes. And then they supplemented it with the OncoScan assay, which is genome-wide copy number with a high resolution in 900 cancer genes. When they first ran only a somatic mutation panel alone, they found that only 35% of the cases had an actionable mutation-based alteration, which is a little bit disappointing to the oncologist, because they now struggle with what to do with the balance 65% of the cases where they did not find any actionable mutations. However, when they added OncoScan, what they saw was the number of patients with an actionable alteration increased from 35% to 80%. And I'm going to share some case examples where this actually happened. This was a patient with a single KRAS G12D mutation. This is a metastatic colorectal cancer. It had a 50% tumor content by sequenome. And what they found when they ran OncoScan was an actionable focal CCND1 gain. Here is an example where they found no somatic mutation. So this is an example which falls in that 65% bucket where they found no actionable mutations. However, they did find a 7-copy CCND1 amplification on OncoScan, which is targetable. Another case here where they found a KRAS 12C mutation. It's a metastatic colorectal cancer. And what they found was a focal CDK6 gain of 7 copies, which gives them one more target to look at in this relapsed cancer patient. Here is a lung adenocarcinoma patient, relapsed cancer case. The oncologist is really looking for what else can be done to help this patient. Ran the sequenome, 0 mutations. But interestingly, even though microarrays typically don't pick up balanced translocations, in this case, they picked up a ROS1 imbalanced translocation. They picked up the ROS1 translocation because an amplification and a deletion event with a break point between the two suggested a ROS1 translocation on this particular patient, which, as we all know, is, here is the evidence. It shows that it defines a unique molecular class of lung cancers and is highly targetable. And here are the list of clinical trials that are recruiting patients with a ROS1 translocation. One more example of lung adenocarcinoma with no mutations. And this patient ended up having an FGFR1 amplification that is druggable. There are several clinical trials that are enrolling patients with FGFR1 amplification. Lung carcinoma, again, no mutations, very abnormal. The first thing that a lot of the labs want to report out, which is completely missing from panel-based approaches, is a whole genome profile. So you get two things out of OncoScan. The first thing that labs are reporting or want to report is the whole genome view. So what you see here is when you see these sharp inflections above the zero line, there are amplifications, and what you say below the zero line are deletions. And what you're seeing here are the chromosomes lined up from 1 through X and Y. So you get two pieces of information. First, you get information to send back to the oncologist that this is a very unstable genome, which in and of itself is a prognostic indicator. And the second thing is, in this particular sample, there were no mutations found. However, this sample did have two druggable copy number alterations, a MET amplification as well as a HER2 amplification. And I'll end with one important real case study on a sarcoma sample from Yale, where this patient had already undergone treatment, you can see here, and they wanted to see if they could get anything else to help this patient from OncoScan. Again, you can see that this is a highly genomically unstable sample, which was important information in and of itself in terms of prognosis. But in terms of actionability, there were two druggable alterations found here on PDGFR-A and another one right next to it. And that gave two options to the oncologist in terms of imatinib or sorafenib. So this oncologist was absolutely delighted that they had two options for these very advanced cancer and are now submitting additional sarcoma cases to see whether they can find actionable copy number, especially in mutation negative samples. And so we get asked this question a lot, that oncologists are very busy people. So how are we going to enable them to get and they are not interested in getting copy number calls and mutations. So how can we give them a report which actually tells them what therapy are available to them? We have a partnership with a company called CollabRx, which can now pull DNA somatic mutation VCF files from any source, including next-gen somatic mutation panels, pull copy number data from OncoScan, and generate a single report outlining therapy options, actionable therapy options in a single report to the lab director, who can then customize it and make it available to the oncologist end user. So now, with the OncoScan providing the copy number component and CollabRx providing the reporting component, there is now a full solution available for oncologists for genomic profiling of actionable alterations. I will stop here, and I think a lot of people joined late. So in summary, what I would like to say is that I would encourage you to go and read the Ciriello Nature paper that defines a class of tumors called copy number tumors that are driven by copy number changes and much less so by somatic mutations. These C-class tumors are ovarian cancer, breast cancer, squamous cell lung cancer, head and neck and prostate cancers. And this really emphasizes the need for researchers to look for copy number changes in these tumors. OncoScan, based on the MIP technology, is especially optimized to look at small fragments of DNA. It only looks at 40 base pairs, and therefore optimized for a whole genome copy number detection from degraded samples, including archived samples. The OncoScan kit has been run on over 12,000 samples and has generated several publications, over 100 in the last 2 years alone, demonstrating a 93% pass rate in FFPE. In addition to research applications, about 30 CLIA labs in the US are adopting OncoScan. Memorial Sloan Kettering, Fox Chase, Seattle Cancer Care Center have gone live. New York State clearance has been received for OncoScan FFPE assay kit to be used as a test. And the way routine use labs are using OncoScan is to combine the mutation data from their NGS panels with copy number data from OncoScan and to deliver a single seamless report on actionable alterations to the end user oncologist. So I want to remind the audience that there is another paper by Frampton et al. That shows that copy number alterations are as actionable copy number alterations are as frequent as actionable somatic mutations in the clinical setting. So the Frampton paper says that actionable copy numbers are found in 41% of the time compared to somatic mutations found 38% of the time in a cohort of 3,000 clinical samples. And also the number of actionable alterations is quite large. It's about at least 23 genes as defined by the Ciriello paper. So with that, I'll stop, and I'm happy to take any questions. I can start with the question that came in through the Q&A. It says, I thought that the OncoScan FFPE kit calculates the CNV in the tumor portion. Does this mean that the algorithm can tell us what the % tumor content of the sample is, as in normal versus tumor cells of the FFPE specimen? That is correct. For the vast majority of the samples that we run, about 80% of the samples, we have a solid tumor algorithm that looks at the fraction of diploid versus non-diploid regions genomically and is able to calculate, therefore, from a genomic perspective, the % aberrant cells or the % tumor cells in the sample and give that number to you. So a typical output would say sample A, it will say % aberrant cells 60%, and then it will give you a copy number value, an integer copy number value of 6 that represents the copy number in the tumor portion only. So this, in our experience, has worked in about 80% of the samples. However, when the samples are very heterogeneous and contain too many clones, then to assign a single tumor burden value, a genomic tumor burden value across the sample is not possible, and there you get a fractional call that represents the average copy number between the tumor and the normal portion. What our users do is, if they have an independent estimate of tumor burden from a pathologist, we have a tool where they can put in that tumor burden estimate in the calculator and can estimate and therefore still arrive at the copy number estimate in the tumor portion only. I don't see any other questions at this time. So at this time, I would like to thank you very much. My name is Padma Sundar. You can feel free. My name is. Let me see if I can share my email address. You're welcome to go to our website and type in OncoScan to learn more about the publications and the MIP assay. And I thank you very much for participating in today's call. Thanks a lot.